I'm Sindhu. I'm a fifth year PhD student at the Epithelial Mechanobiology lab. Here I'm interested in studying the biophysics of epithelial tissues. So these are tissues that line the surfaces of our organs, and the cells are very tightly packed here. So they are all tightly packed, they have very interesting physical properties which resemble classes. For example, they can move together as groups. So my main work is looking at how the physical properties that come from crowding and that can interact with the biological properties of the cells. Generally, when particles are sparse, they can move freely, but as they get more and more crowded, movement of particles are restricted by their neighbors. So the dynamics slows down drastically and they show glassy dynamics. One important feature of glassy dynamics is dynamic heterogeneity. It means that there are groups of fast particles and groups of slow particles which are clustered together in space. But what happens in a system where there is crowding as well as activity, meaning that the individual particles are themselves motile? On the one hand, activity tends to fluidize the system, but on the other hand, crowding pushes it towards glassy dynamics. So, do epithelial monolayers show glassy dynamics or fluid-like behavior? We found that although epithelia have active cells, they show glassy dynamics. This is because the fast group of cells here have more actin in the form of stress fibers, whereas the slow group of cells have more cortical actin architecture. This shows that there is biochemical a crosstalk between cell mechanics and biochemical signaling. So, mechanochemical feedback loops can stabilize the fast and slow groups of cells, and epithelia show glassy dynamics although they are active. And apart from that, I'm also looking at how this crosstalk affects cancer initiation and the cysts that form in kidney cystic diseases.